My name's Dr. Katie Lunnon. I'm a senior lecturer at the University of Exeter Medical School, and my research looks at the role of epigenetic mechanisms in dementia, and I recently won the Alzheimer's Research UK Early Career Investigator of the Year award. Essentially, we know that in diseases such as Alzheimer's disease that it's not entirely caused by genetic changes in individuals, and that environmental influences might have an impact. So we look at something called epigenetics, which is essentially the addition of chemical tags to the DNA which can turn genes on and off. And so we look at brain samples from people with dementia, see if we can see these in regions of the brain that might have high levels of Alzheimer's pathology. At the moment, we don't know what actually causes dementia, so it's particularly important to try and identify new causes. One of the really interesting things about identifying epigenetic changes is if we can show that they're causative in disease, these are actually potentially reversible. The impact of our work so far has been, we published the first Epigenomewide association studies, which essentially are studies where you look at levels of a chemical tag across the genome. So we looked at the levels of a chemical tag called DNA methylation across a group of 120 people with various stages of Alzheimer's disease in various regions of their brain. Interestingly, at the same time, another group also were doing a similar study, and they were looking at around 700 people just in one brain region with various degrees of Alzheimer's disease. And what was really interesting was we both found the exact same gene as one of our most altered genes in disease called ANC1. What's really interesting is since even whichever brain cohort we look at, we actually see increased levels of the DNA methylation tag on this gene in people with Alzheimer's disease compared to control, which I think shows quite how robust and consistent the epigenetic changes are in Alzheimer's disease brain. One of the issues at the moment is we don't know if these changes are a cause of the disease or actually a consequence of the disease, and that's probably an avenue that our research will be following over the next few years. Our end goal is to try and identify new molecular mechanisms that might be involved in disease, which might help us then identify new drug targets for pharmacological intervention. Another key aim of ours is to look in blood samples from people with dementia, try and identify biological markers or biomarkers in blood, which could help to diagnose the disease a lot earlier. One of the issues with Alzheimer's disease at the moment is that we don't know what causes the disease, and we're not diagnosing it early enough, and that current treatments do not only treat the symptoms but do not actually reverse the pathology. So if we were able to identify new mechanisms that are involved in disease and we're able to diagnose the disease earlier, then it might have a significant impact on people's lives.